So our next presenter is Kiara Kidman. Her project is supervised by Ricardo, Mancera and Cara Kreck from the Curtin Medical School.
Hi everyone, my name is Kiara and my project was visualising how the proteins responsible for type 2 diabetes look in 3D. The aim of this project was to highlight how advanced visualisation techniques provide the best environment to study a protein's molecular structure and the interactions which occur within and between these molecules. To achieve this, I used immersive stereoscopic three-dimensional visualizations of a protein known as amylin, which is responsible for type 2 diabetes. Now let's begin with a brief introduction into proteins. Atoms can interact with each other to form amino acids. The image on the far right shows the general structure of the different types of amino acids. Now, amino acids are just the building blocks of proteins. They are joined together by what is called a peptide bond to produce a long chain of these amino acids, as represented by the middle figure. These chains of amino acids are what we call a protein. The atoms making up a protein can be arranged differently in three-dimensional space and are able to transition from one arrangement to another. These arrangements determine the shape of the protein that the protein will make. These shapes are referred to as a protein's secondary structure and are commonly used to visualize the protein. A common secondary structure produced by proteins is an alpha helix, which you can see in the above coiled rod-like structure in blue. Another secondary structure is called a beta strand, which is just a long chain of amino acids, which represented in an extended conformation, seen in this image over here. Now, a beta sheet is just a collection of various amino acids that are joined together. This joining together produces them to become stacked on top of each other to form a sheet-like structure seen in the purple image here. Many naturally occurring proteins are involved in the development of diseases. Type 2 diabetes is a chronic disease that is affecting an increasingly large proportion of the world's population. It is characterized by persistent high blood sugar levels that is caused by the impaired insulin production, secretion and function. Now, at the molecular level, the cells within the pancreas, called beta cells, are responsible for producing insulin and amylin, which both aid in regulating body blood sugar levels. The amylin protein is able to regulate these levels by slowing down the digestive system. Amylin is what we call an intrinsically disordered protein. This means it lacks a well-defined structure, resulting in it having a flexible conformation. Additionally, the amylin proteins are able to rapidly join together in a process known as aggregation. Now, aggregation is when a single protein, referred to as a monomer, joins together with another monomer to form what we call a dimer. These dimers can then go on to join with other molecules to produce larger, more complex versions of them, known as oligomers. In the case of amylin, this rapid aggregation results in the formation of toxic oligomers. These toxic proteins are responsible for pancreatic beta cell death. Now, remembering that these cells are responsible for producing insulin, therefore, their death results in impaired insulin production and regulation, which is the cause of type 2 diabetes. To further implicate amylin's role in the development of type 2 diabetes, researchers found toxic deposits in the pancreas of 90% of the type 2 diabetic patients they autopsied. It is crucial for researchers to be able to investigate the structural arrangements of amylin that causes aggregation into these toxic proteins. Unfortunately, due to amylin's disordered nature and rapid aggregation, it is extremely challenging for researchers to use experimental methods to identify key structural conformations occurring within a single molecule of amylin. To overcome these challenges, molecular simulations can be used. These are advanced computational methods that often require the use of supercomputers, such as PAUSI. These simulations are able to capture in intense detail the behaviour of complex molecules in physiological conditions. In order to sufficiently sample amylin structures, I ran 3 simulations concurrently with each other to sample the different secondary structures amylin samples. The first simulation was set to sample alpha-based structures, the second beta, and the third was set to sample both alpha and beta. Now, in order to maximise the usefulness of molecular simulations, it is critical to use sophisticated visualisation software to produce high-quality, realistic representations of these simulated biomolecules. Interactive graphical representations of molecular structures have been instrumental in aiding our understanding of complex biomolecular systems and have also been used to communicate these important discoveries to the population. For this project, I used a program called Molecular Dynamics Viewer, MDV for short. It was used due to its ability to be able to allow the user to view molecules in stereoscopic 3D, which provides us with an immersive, interactive experience for the user, which is a feature that definitely goes beyond the standard capabilities of standard visualization tools. It also provides a graphical user interface, which allows the user to be able to upload their molecular structure, modify the atoms, the size of the atoms and the bonds, and to select and isolate specific amino acids of interest. MDV also incorporates a third-party tool called Stride, which calculates and displays the protein's secondary structures. MDV was specifically designed for projecting molecular structures onto the HIVE Cylinder display, which provides us with a wide 180-degree viewing angle. To enhance MDV's performance, we further refined the amylin simulations to create detailed still and animated visuals of amylin's structural transformations. The figures on the bottom of this slide were used to identify the key time points within the simulation where amylin was demonstrating the desired secondary structures of our interest. These time points were then used to produce static representations. During molecular simulations, the protein is surrounded by water and iron molecules, which we decided to omit from the final visuals so that we could focus on amylin itself. For the dynamic visualizations, we select the specific regions within the simulation where amylin was exhibiting the highest levels of the desired secondary structure. For further, we eliminated any rapid movements occurring within these dynamic visualizations to ensure that the resulting animations were smooth and clear for viewers to see. In order to get the full experience of the visualization capabilities of MDV, I will be demonstrating my project in further detail on the cylinder display during the allocated time. However, for the purpose of this presentation, I have produced a series of snapshots of the static representations of the amylin protein using MDV. The above images capture the static representation of the first simulation. As you can see on the far right image, amylin is displayed using a ball and stick model, with the balls representing atoms and the sticks representing bonds. This style of visualization is commonly referred to as CPK. The middle static image shows us the secondary structure produced by amylin when the atoms are arranged in this arrangement. This means that the atoms you see in the far right image are arranged in a way which produces an alpha helical structure seen in the last two images in blue. To highlight how visualizations can be further modified to extract insightful meaning, I added the last image to the left over here, where I specifically selected only the amino acids from 20 to 29 to be displayed in a CPK model. The reason I chose to display these specific amino acids is because they have been implemented and identified as the key amino acids which cause amylin to aggregate into these toxic proteins. As you can see, these amino acids are within the helical structure. These images are of the second simulation where amylin is exhibiting beta structures. The beta structures can be seen in these images to the left of me as represented by the large red arrows. Remembering that beta sheets are formed by beta strands stacking on top of each other. Additionally, you can see another type of beta secondary structure known as a beta turn in these images, which is represented in the bright yellow. Again, the last image was produced with the same amino acids regions being highlighted, showing us the ability of these amino acids to be able to transition from one type of secondary structure to another. Now, two static representations were produced for the third simulation, which as previously mentioned was set to sample both alpha and beta conformations. The images on the screen show at the time point in the third simulation where amylin exhibited the highest levels of alpha secondary structures, which can be seen by the blue helicals in the images. Additionally, in these images, you can also see beta structures being sampled that was exhibited by amylin at this point in the simulation. On this next slide, you see the portion within the third simulation used to produce these second static images. These images can clearly see the beta sheet structures represented in red and the beta turns in yellow. Interestingly, no alpha structures were demonstrated at this time point in the third simulation, unlike with the previous slide where you can see both structures being produced. The images you see today have been static representations of amylin's secondary structure. However, you should keep in mind that in real life, amylin is continuously transitioning from one structural arrangement to another within nanoseconds of each other. Because of this fact, I have produced dynamic visualizations to be able to visualize the rapid structural transformations. I have not included these animations into the presentation, as it would not do them justice, as they were specifically produced to be viewed on the Curtin cylinder. During the demonstration, I'll be visualizing the static structures produced from the molecular simulations of amylin. These static renderings, when viewed on the cylinder, will enable the viewer to be able to go inside the molecule and be immersed in an atomic environment, which allows us to further investigate in greater detail amylin structures in 3D. On top of these static images, dynamic visualizations will also be shown. This is where you'll be able to watch as the amylin protein transitions from one conformation to the next. Additionally, using previously simulated data, I was able to produce dynamic simulations of two single amylin molecules interacting with each other to form a dimer. I hope these demonstrations are able to show you the importance of how high-quality visualizations aid in our understanding of proteins involved in the development of diseases such as type 2 diabetes. Lastly, I'd like to thank all my supervisors for not only providing me with this opportunity, but for their continued support. Any questions?